Welcome to our deep dive into the fascinating world of isomerism. Have you ever wondered how two compounds can share the same chemical formula yet behave so differently? That's the magic of isomerism. In this video, we'll explore what isomerism is, why it matters in chemistry, and how it influences everything from the medicines we take to the flavors we taste. Get ready to unravel the mysteries behind molecular structures and discover how small changes can make a big difference. Let's embark on this molecular journey together, starting with the basics of isomerism and its significance in the scientific world. Isomers are molecules that have the same molecular formula but different arrangements of atoms. This means that even though they contain the same types and numbers of atoms, their structures are distinct, leading to different physical and chemical properties. Imagine two houses built with the same number of bricks, but arranged in completely different ways. Each house would look and function differently. In chemistry, this concept is crucial because it helps explain why substances with identical formulas can have unique behaviors. Understanding isomers is the first step to grasping the broader concept of isomerism. Isomerism isn't just a single concept. It branches into several types, each with its own unique characteristics. The two main categories are structural isomerism and stereoisomerism. Structural isomers differ in the connectivity of their atoms, while stereoisomers have the same connectivity but differ in the spatial arrangement of atoms. These distinctions are fundamental in chemistry, as they help scientists classify and understand the diversity of molecular structure. Throughout this video, we'll break down each type, providing clear examples and explanations to make these concepts easy to grasp. Start with structural isomerism. Structural isomers, also known as constitutional isomers, have the same molecular formula but their atoms are connected in different ways. This can lead to variations in the carbon chain, the position of functional groups, or even the presence of different functional groups altogether. For example, butanol and isobutanol both have the formula C4H10O, but their structures and properties are distinct. Structural isomerism is a key concept in organic chemistry, as it explains the diversity of organic compounds and their behaviors. Anisomerism is a type of structural isomerism where compounds differ in the arrangement of their carbon skeletons. For instance, pentane, isopentane, and neopentane all have the formula C5H12, but their carbon chains are arranged differently. This leads to variations in their boiling points, melting points, and other physical properties. Chain isomerism is commonly observed in alkanes and is an important concept for understanding the diversity of hydrocarbon. By rearranging the carbon backbone, chemists can create molecules with unique characteristics, all from the same set of atoms. Position isomerism occurs when the functional group or substituent is attached to different positions on the same carbon chain. For example, 1-butanol and 2-butanol both have the formula C4H10O, but the hydroxyl group is attached to different carbons. This small change can significantly impact the compound's reactivity and properties. Position isomerism is especially important in organic synthesis, as the location of a functional group can determine how a molecule interacts with other substances. It's a subtle but powerful way that molecules can differ. Functional group isomerism arises when compounds have the same molecular formula but different functional groups. For instance, ethanol and alcohol and dimethyl ether and ether both have the formula C2H6O, yet their properties and uses are vastly different. This type of isomerism highlights how the arrangement of atoms into different functional groups can create entirely new substances. Functional group isomerism is crucial in both organic and medicinal chemistry, as it explains why molecules with the same atoms can have completely different effects. Tautomerism is a less common but fascinating type of structural isomerism. It occurs when compounds have the same molecular formula and functional group, but the alkyl groups attached to the functional group differ. For example, diethyl ether and methylpropyl ether both have the formula C4H10O, but their alkyl groups are arranged differently around the oxygen atom. Tautomerism is often seen in ethers, amines, and other compounds with divalent atoms. This subtle rearrangement can lead to differences in physical and chemical properties. Tautomerism is a dynamic form of isomerism where compounds exist in equilibrium between two structural forms, usually involving the movement of a hydrogen atom and a double bond. A classic example is keto-enol tautomerism, where a compound can switch between a keto form and an enol form. This process is reversible and plays a significant role in biochemical reactions and organic synthesis. Tautomerism demonstrates how molecules can be flexible, adapting their structure under different conditions to achieve stability or reactivity. Moving beyond structural isomers, let's explore stereoisomerism. Stereoisomers have the same molecular formula and connectivity, but differ in the spatial arrangement of their atoms. This means the atoms are connected in the same order, but their three-dimensional orientation is different. Stereoisomerism is especially important in biology and medicine, as the 3D shape of a molecule can determine how it interacts with enzymes, receptors, and other biological molecules. Let's break down the main types of stereoisomerism and see why they matter. Geometric isomerism, also known as cis-trans isomerism, occurs in compounds with restricted rotation, such as those containing double bonds or ring structures. In cis-isomers, similar groups are on the same side of the double bond, while in trans isomers they're on opposite sides. This seemingly small difference can have a big impact on the compound's properties, such as boiling point, solubility, and biological activity. Geometric isomerism is commonly seen in alkenes and some cyclic compounds, making it a key concept in organic chemistry. Optical isomerism is a fascinating type of stereoisomerism, where molecules are non-superimposable mirror images of each other, much like your left and right hands. These isomers are called enantiomers. Optical isomers rotate plane polarized light in different directions, one to the right dextrorotatory and one to the left levorotatory. This property is crucial in pharmaceuticals, as one enantiomer of a drug may be therapeutic, while the other could be inactive or even harmful. Optical isomerism highlights the importance of molecular symmetry and chirality. Chirality is the property of a molecule that makes it non-superimposable on its mirror image. Molecules with a chiral center, usually a carbon atom bonded to 4 different groups, can exist as 2 enantiomers. These enantiomers have identical physical properties except for the direction in which they rotate plane polarized light and their interactions with other chiral substances. Chirality is a central concept in organic chemistry and biochemistry, influencing everything from drug design to the flavors of food. Diastereomers are stereoisomers that are not mirror images of each other. Unlike enantiomers, diastereomers have different physical and chemical properties, such as melting points and solubilities. They often arise in molecules with multiple chiral centers. For example, tartaric acid has 2 chiral centers and can form several diastereomers. Understanding diastereomers is important in chemistry, as their distinct properties can be exploited in separation techniques and synthesis. Meso compounds are a special case in stereochemistry. They contain multiple chiral centers but are superimposable on their mirror images due to an internal plane of symmetry. As a result, meso compounds are optically inactive. They do not rotate plane polarized light. This unique property makes them stand out among stereoisomers. Meso compounds often appear in molecules like tartaric acid, where symmetry cancels out optical activity. Recognizing meso compounds is essential for understanding the full spectrum of isomerism. Isomerism plays a critical role in the pharmaceutical industry. Many drugs exist as isomers, and often only one isomer is therapeutically active. The other may be less effective or even cause side effects. For example, the drug thalidomide exists as 2 enantiomers. One treats morning sickness while the other causes severe birth defects. This highlights the need for precise control over isomerism in drug design and manufacturing, ensuring safety and efficacy for patients. Isomerism isn't just important in medicine, it also affects the flavors and aromas of food. For example, the 2 enantiomers of carvone smell completely different. One gives spearmint its characteristic scent while the other smells like caraway. This is because our taste and smell receptors are chiral, responding differently to each isomer. Understanding isomerism helps food scientists create flavors and fragrances that appeal to our senses, making our culinary experiences more enjoyable. Nature is full of isomers, from the sugars in our bodies to the pigments in plants. For instance, glucose and fructose are structural isomers with the same formula but different structures, leading to different roles in metabolism. Similarly, the color of flowers and fruits often depends on the arrangement of atoms in pigment molecules. Isomerism allows for diversity in biological systems, enabling organisms to adapt and thrive in different environments. Isomerism also influences the properties of materials. For example, polymers made from different isomers can have vastly different strengths, flexibility, and melting points. The arrangement of atoms and monomers determines how the polymer chains interact, affecting the material's performance. This knowledge is used to design plastics, fibers, and other materials with specific properties for various applications, from clothing to aerospace engineering. You encounter isomerism more often than you might think. From the sweeteners in your drinks to the fragrances in your soap, isomers are everywhere. Even the gasoline in your car contains different isomers of hydrocarbons, affecting its performance and efficiency. Understanding isomerism helps us appreciate the complexity and versatility of the molecules that make up our world, showing how small changes at the atomic level can have big impacts on our daily lives. The concept of isomerism was first recognized in the early 19th century, when chemists noticed that some compounds shared the same formula but had different properties. Friedrich Wörthler's synthesis of urea in 1828 was a turning point, showing that organic compounds could be created from inorganic materials. This discovery challenged existing theories and paved the way for the development of modern organic chemistry. The study of isomerism has since become a cornerstone of chemical science. Isomerism can influence how molecules react with each other. Structural isomers may undergo different reactions due to the arrangement of their atoms, while stereoisomers can interact differently with enzymes or catalysts. This is especially important in organic synthesis, where selecting the right isomer can lead to higher yields or desired products. Understanding isomerism allows chemists to predict and control chemical reactions more effectively. Analytical chemists use various techniques to distinguish between isomers. Methods like chromatography, spectroscopy, and crystallography can separate and identify isomers based on their unique properties. This is vital in fields like pharmaceuticals, where purity and identity are crucial. By understanding isomerism, scientists can ensure the safety and effectiveness of chemical products, from medicines to industrial chemicals. Isomerism also plays a role in environmental science. Some pollutants exist as isomers, with one form being more toxic or persistent than another. For example, certain pesticides have isomers that differ in their environmental impact. Understanding these differences helps scientists develop safer chemicals and assess environmental risks more accurately. Isomerism thus contributes to protecting our planet and promoting sustainability. Biochemistry, isomerism is fundamental to life. Many biomolecules, such as amino acids and sugars, exist as specific isomers. For example, only L-amino acids are used to build proteins in living organisms, while D-amino acids are rare. The specificity of enzymes for certain isomers ensures the proper functioning of metabolic pathways. Isomerism thus underpins the complexity and precision of biological systems. Industrial chemists exploit isomerism to create products with desired properties. For example, the octane rating of gasoline depends on the types of hydrocarbon isomers present. High-octane fuels contain more branch isomers, which burn more efficiently. Similarly, the production of plastics, solvents, and other chemicals often involves selecting or synthesizing specific isomers to achieve optimal performance. Isomerism is thus a powerful tool in industrial innovation. Organic synthesis often relies on controlling isomerism to produce specific compounds. Chemists use catalysts, reagents, and reaction conditions to favor the formation of one isomer over another. This is crucial in the synthesis of pharmaceuticals, agrochemicals, and materials. By mastering isomerism, chemists can design efficient synthetic routes and minimize unwanted byproducts, making chemical processes more sustainable and cost-effective. Polymers can exhibit isomerism in the arrangement of their monomer units. For example, isotactic, syndiotactic, and atactic polymers have different spatial arrangements of side groups, affecting their crystallinity and mechanical properties. This allows manufacturers to tailor polymers for specific uses, from flexible packaging to strong engineering plastics. Understanding isomerism in polymers is key to developing advanced materials for modern technology. Ordination compounds, often found in transition metal chemistry, can also exhibit isomerism. Ligands can attach to the central metal atom in different ways, leading to structural and geometric isomers. For example, cisplatin and transplatin are geometric isomers with very different biological activities. Cisplatin is a powerful anti-cancer drug, while transplatin is not. This highlights the importance of isomerism in inorganic chemistry and medicine. Aromatic compounds, such as benzene derivatives, can also show isomerism. For example, ortho, meta, and para isomers refer to the positions of substituents on the benzene ring. These positional isomers have different chemical reactivities and physical properties, which are important in the synthesis of dyes, pharmaceuticals, and polymers. Understanding isomerism in aromatic compounds is essential for designing functional molecules in various industries. Carbohydrates like glucose and fructose are classic examples of isomerism in biochemistry. These sugars have the same molecular formula but different structures, leading to different metabolic pathways and functions in the body. Additionally, carbohydrates can exist as stereoisomers, such as D and L-glucose, which are processed differently by enzymes. Isomerism in carbohydrates is fundamental to nutrition and energy metabolism. Amino acids, the building blocks of proteins, also exhibit isomerism. Most naturally occurring amino acids are L-isomers, which are incorporated into proteins by ribosomes. D-amino acids are rare in nature but can be found in some bacterial cell walls and antibiotics. The distinction between L and D isomers is crucial for protein structure and function, highlighting the importance of isomerism in biology. Nucleic acids, such as DNA and RNA, contain sugar isomers that determine their structure and function. For example, DNA contains deoxyribose, while RNA contains ribose. Both are isomers with different properties. The arrangement of these sugars affects the stability and reactivity of genetic material, influencing how information is stored and transmitted in living organisms. Isomerism in nucleic acids is thus essential for genetics and molecular biology. Lipids, including fats and oils, can also exhibit isomerism. For example, cis and trans isomers of fatty acids have different effects on health. Cis fats are generally considered healthier, while trans fats are associated with increased health risks. The arrangement of hydrogen atoms around double bonds in fatty acids determines their physical properties and impact on the body. Understanding isomerism in lipids is important for nutrition and health science. Some vitamins exist as isomers, with only one form being biologically active. For example, vitamin A has several isomers, but only retinol is essential for vision and health. The other isomers may have different or no biological activity. This specificity underscores the importance of isomerism in nutrition and the design of dietary supplements, ensuring that we get the right form of each vitamin. Hormones, the chemical messengers of the body, can also exist as isomers. For example, adrenaline and noradrenaline are structural isomers with different physiological effects. The arrangement of atoms in these molecules determines how they interact with receptors and trigger responses in the body. Isomerism in hormones is crucial for understanding how the body regulates processes like stress, metabolism, and growth. Perfume industry relies heavily on isomerism to create unique scents. Isomers of the same compound can have completely different odors, allowing perfumers to craft a wide range of fragrances. For example, the isomers of ionon smell like violets or raspberries depending on their structure. Understanding isomerism enables the creation of signature scents that appeal to our senses and emotions. Dyes and pigments often rely on isomerism to produce different colors. The arrangement of atoms in a dye molecule can affect how it absorbs and reflects light, resulting in a spectrum of colors. This is important in industries ranging from textiles to art supplies. By manipulating isomerism, chemists can create vibrant and stable colors for a variety of applications. Agrochemicals, such as pesticides and herbicides, often exist as isomers with different levels of effectiveness and safety. Selecting the right isomer can improve crop yields while minimizing environmental impact. Regulatory agencies require detailed analysis of isomers to ensure that only the safest and most effective forms are used in agriculture. Isomerism thus plays a key role in sustainable farming. Isomerism can influence the performance of energy storage materials such as batteries and fuel cells. The arrangement of atoms in electrode materials can affect their conductivity, stability, and capacity. By designing materials with specific isomeric forms, scientists can develop more efficient and durable energy storage solutions, supporting the transition to renewable energy sources. Some isomers are more easily broken down by environmental processes, making them preferable for use in products that may end up in the environment. By selecting isomers that degrade more readily, chemists can design safer chemicals that minimize pollution and support environmental remediation efforts. Isomerism thus contributes to greener and more sustainable chemistry. Isomerism isn't limited to Earth, it's also found in space. Astronomers have detected isomers of organic molecules in interstellar clouds, providing clues about the origins of life and the chemistry of the universe. Studying isomerism in space helps scientists understand how complex molecules form and evolve in extreme environments, expanding our knowledge of chemistry beyond our planet. Artists and designers sometimes draw inspiration from isomerism, using the concept of different arrangements to create visually striking works. The idea that small changes in structure can lead to big differences in appearance is mirrored in art, where composition and arrangement are key. Isomerism thus bridges the gap between science and creativity, showing how molecular concepts can influence aesthetics. Research into isomerism continues to advance, with scientists discovering new types of isomers and developing innovative ways to control molecular structures. These breakthroughs have the potential to revolutionize fields like medicine, materials science, and environmental protection. As our understanding of isomerism grows, so too does our ability to harness its power for the benefit of society. Conclusion: isomerism is a fundamental concept that shapes the world around us. From the medicines we take to the materials we use, isomers influence countless aspects of our lives. By understanding isomerism, we gain insight into the diversity and complexity of chemistry, unlocking new possibilities for innovation and discovery. Thank you for joining us on this journey through the world of isomerism. Stay curious and keep exploring the wonders of science.